have transcription, where the DNA is transcribed into mRNA. We start with the DNA in the nucleus. These DNA strands contain genes that code for various amino acids. In order to translate this into RNA, we need an enzyme. This is where RNA polymerase II comes in. The DNA is split in half, and the bases are exposed. Then, the RNA polymerase starts to move down from the 5' end to the 3' end in an elongation. It builds the RNA off the template strand, which contains bases complementary to the mRNA forming on it. The other strand is the coding strand, which is similar to the mRNA, although it is not directly involved in transcription. Keep that in mind. Thymines turn into uracils. Afterwards, termination begins, where RNA polymerase is removed and the DNA is rebound.
After the termination coding sequence for the base pairs, in this case UAA, we begin the process of mRNA editing. The start of the process for mRNA editing begins when enzymes alter both ends of the pre-mRNA strand. To begin, the enzymes add a modified guanine nucleotide to the 5' end of the pre-mRNA strand. The addition of the guanine nucleotide is known as the 5' cap. Literally, on the other hand, 50 to 250 adenine nucleotide is added to the 3' end. We call this the poly A tail. After the 5' cap and the poly A tail are added to the pre-mRNA strand, assuming that all of these are extrons, this mRNA strand is ready for translation.
mRNA now arrives in the cytoplasm. The 5' cap and poly A chain protect the mRNA until it reaches the ribosome. The small ribosomal unit attaches to the mRNA. This stage is known as initiation. Then a tRNA attaches to the mRNA using corresponding anticodons with the codons, and has a methionine that corresponds with the start codon AUG. The large ribosomal unit then attaches to the top. There are 3 sites for the tRNAs. There's the E, P, and A sites that are formed after the large subunit attaches at the top. Once the tRNA and mRNA are bonded, the amino acid detaches with the help of a transferase and moves along to the next tRNA. This process is known as elongation. Once that happens, the next tRNA bonds its anticodons with the corresponding codons and moves along. The amino acids begin to make a polypeptide chain and continue along their path until finally a stop codon comes along. Once this happens, the last tRNA is discarded and the amino acid chain becomes a protein. This stage is known as termination, and this finishes with the protein.